Afternoon to one and all. Myself Dr.J.HEMAPRIYA from Department of Microbiology. Today I am going to discuss about the topic TI plasmid. So what is TI plasmid? Plasmids are nothing but extra chromosomal self-replicating body which is present in most of the bacteria, and it gives an extraordinary power to the bacteria that we know already. And what are TI plasmids? TI means tumor inducing. Okay, so TI plasmids are called tumor inducing plasmids. So TI plasmid based vectors are having very high efficiency. That's why we call them as high efficiency vectors. And it was the main aim of developing this TI plasmid is to introduce new genes into the plants actually. So Agrobacterium tumefaciens, actually this is a bacteria which is a gram-negative bacteria, and it is a soil bacteria. This Agrobacterium tumefaciens only possesses this TI plasmid, and this bacteria actually causes a disease called crown gall disease in plants, in which the cells grows in an undifferentiated manner, because we all know what is a tumor. Tumor means undifferentiated growth of cells in an uncontrollable manner. Okay, right. So which results in the formation of a tumor. So this Agrobacterium tumefaciens, it's a gram-negative soil bacteria, and it possesses the tumor causing ability, and because of the presence of this TI plasmid. Alright. So what is the structure of, how do we define the structure of TI plasmid? Means it has 196 genes, including one structural RNA which encodes for 195 proteins. And this plasmid actually has a very important region called the tDNA region, which is about 20 kilobase pair in size. And this is the region, actually this is the unique region which is going to get transferred to the plant cell, and it gets integrated into the plant DNA. So this is the most important portion of the TI plasmid actually. And even though after the transmission also, the most unique feature of this tDNA is it stably gets transmitted through the divisions, like when the plant cell undergoes mitosis and meiosis. Even after the mitosis and meiosis also, it does not get disturbed, and this the tDNA gets successfully transmitted from one generation to the next generation of the cell actually. So that is a unique feature. So this transfer of tDNA from Agrobacterium to plant cell, it depends on the 25 base pair repeated sequence which is present at the both the sides of the tDNA. Actually, when I discuss about the structure of TI plasmid, you will come to know what are these repeated sequences on both the sides of the tDNA. Okay, then the tDNA carries an enzyme actually. So it is having enzyme. How it causes tumor? How the TI plasmid is having the ability to cause tumor means this tDNA carries enzymes for certain kind of plant hormones which are nothing but the auxins and cytokinins. So we all know that auxins and cytokinins are nothing but they are all phytohormones which can cause the abnormal cell growth actually. So because of this particular reason only, when tDNA enters the plant cell, a tumor is getting formed actually. So it also carries genes for, there are other type of genes called opine genes, because see the bacteria is going to survive in the plant. So how do the bacteria get food? So bacteria needs food also. So what it will do is, it will carry a gene called opine gene which could able to synthesize its own food actually. So this opine serves as a food for the bacterium. So it's carrying its own tiffin carrier, it's like that. So it is taking the opine, there is an opine gene which is going to act as a food for the bacteria, right? So it goes comfortably, cause disease, sit there, eat very nice food, and then it will grow as a tumor. That's what going to happen here. Okay, so and what the researchers have done is they wanted to use this quality of the TI plasmid in the genetic engineering purpose. So what they did is they did some change in the TI plasmid, and genetically they converted the TI plasmid into a transfer for the use of transferring the genes actually, right? So what they did for this particular conversion is they disturbed the tumor causing gene as well as the opine synthesis gene, and they replaced them with the selectable markers which is conferring the resistance to the antibiotics. And TI plasmid based vectors were also constructed with the following components. So these are the components actually recombinant TA plasmid has. Okay, the first one is origin of replication. So origin of replication is must for any plasmid, that we all know it. So it allows the plasmid to replicate. The second one is, as I said, the tDNA will be accommodated by on both the sides by two borders called right border sequence and left border sequence. Okay, so the right border sequence which is necessary for the transfer of TA plasmid. So the right and left border sequence, they are only the most important part of the TA plasmid, which is going to help the tDNA to get transferred from the TA plasmid, I mean from the bacterium to the plant cell. Okay, and it is also having a multiple cloning site, because we have to insert our own gene of interest, right? So for that purpose, we are going to insert, I mean the researchers have inserted a multiple cloning site also for the ease of insertion of any gene of interest, okay, and between the tDNA border sequences, right? So recombinant genes can very easily get integrated into the tDNA of the TA plasmid, and the plasmid can be used to infect the plant cells. Okay, right. So the infected cells are placed on a culture medium, because this all of us know how to select the recombinant cells, or how to select the recombinant culture cells. The infected cells will be placed on a culture medium which contains growth factor as well as the selectable antibiotics, because we have already inserted selectable markers which are nothing but the antibiotics. So we are going to put it in a medium which contains the antibiotic. As a result of that, all the other cells will die. Only the TA plasmids which harbors the tDNA with the antibiotic markers only can survive in that. Okay, so with this we can able to easily identify the recombinant TA plasmids, and this is the structure of TA plasmid. So here you can very clearly see the structure. So I have given the raw structure, I did not give the genetically modified structure. So here you have the tDNA region. This is the most important region which is going to get transferred to the plant actually. So this is called left border and this is called right border. So in between the right border and left border only, you have the most important region called TA region. Okay, sorry, tDNA region. So in this tDNA region, as I said earlier, you have two important enzymes. I mean two important enzymes which are important for the production of phytohormones called auxins and cytokinin. These are the genes which are responsible for the auxin production and cytokinin. Okay, and here you have the opine gene, and here you have the opine catabolism gene. Here this is origin of replication. This is the virulence gene. Okay, so this is the structure of TA plasmid, right? Okay. So next coming to the transfer DNA, which as I said, this is going to be the most important part of the TA plasmid. We are going to discuss very deeply about the TA plasmid. So the TA plasmid is transferred from bacteria to the host plant's nuclear DNA actually. So the DNA is bordered by 25 base pair repeats on each end actually. As I said, here you have the left border as well as you have the right border. So these are made up of 25 base pairs repeats on either end. Excuse me. So transfer is initiated at the right border. Okay, so you have a right border and left border. But when you take the transfer initiation, that is happening in the right border. Okay, so the transfer gets initiated in the right border and get terminated in the left border. Okay, right. So this requires actually two genes. One is called Vir gene of the TA plasmid. Another one is nothing but a single-stranded DNA actually. So these two are the things which is initiating the right border, which is initiating the transfer of tDNA. So the bacterial tDNA is about 24 base pair long and contains genes that code for the enzymes, that is opines and phytohormones. As I said earlier, the phytohormones are nothing but the cytokinin and the auxin. Okay, right. So by transferring the tDNA into the plant genome, what happens in the plant is the plant completely reprograms its function. Okay, so what do you mean by reprogramming of the function? Nothing but the effect of cytokinin and auxin is going to create an undifferentiated growth of cell which is going to result in a tumor formation. Okay, so that is what is given here. So the plant is getting reprogrammed. A complete function will be reprogrammed as per the instruction of the newly inserted TA plasmid, TDNA region, and as a result the tumor growth will be there. Okay, so the synthesis of plant hormones, auxin and cytokinin, they only initiate the uncontrolled growth of the cell, which results in the formation of tumor actually, right? So tDNA transformation again. So agrobacterium mediated tDNA transfer is widely used in biotechnology because this is the most even in plant biotechnology a wide use is there for this tDNA actually. So in genetic engineering, the tumor promoting and opine synthesis genes will be removed actually, and then it will be replaced with the gene of interest. So as I said, these two genes are not required for the plant actually, because we are going to remove the tumor promoting gene as well as the opine genes, because they are all unwanted things. Instead of unwanted genes, we are going to replace that with gene of our own gene of interest. Like if you want to, for example, if you want to insert a particular character to the plant, okay, so you can take the gene, you can isolate the gene and you can insert that in this place actually. Okay, as well you can insert a selection marker, that is the antibiotic marker actually. So likewise, in genetically engineered TA plasmid will be having the tumor promoting and opine synthesis gene replaced with a selection marker and the gene of interest. Okay, right. So if you talk about the selection markers, what are all the antibiotics which are all generally used for this particular processes? One is neomycin phosphotransferase and another one is you can use hydromycin B also. So these two antibiotics can be used as a selectable marker actually. And now we are going to see about the mechanism of tDNA transformation. So coming to the mechanism of tDNA transformation, the first step is the integration of tDNA into the host genome is the formation, which is going to get started with the nick formation. So what do you mean by nick? Nick is nothing but a gap. Okay, so first a nick will be formed at the right border. So in the earlier slide itself I told you that the transfer of tDNA will start or will get initiated from the right side, from the right border. So if that is the case, the nick will be formed first at the right side and it gets continued towards the left. Okay, it creates a region of single stranded DNA from the left border of the tDNA and over to the right border, and it gets cut off, it gets walled off. Okay, so you have you have now the tDNA portion separately. Okay, and here I have given the stepwise process of schematic representation of the agrobacterium mediated transformation. Here, if you have, if you see this, you have 9 steps actually. Okay, the first step is the production of signal molecules. So I'll just show you with the diagrammatic representation. Here we have 9 steps, and the first step is the production of signal molecules from the wounded plant cell, right? So here you can see this is the first step, right? From the plant cell. See, one thing I want to mention here, generally a normal plant, okay, will not invite agrobacterium tumefaciens, okay. A wounded plant, for example, because of soil erosion or because of some human interrupted some kind of wound formation to the plants, because of all these things, or animals, because of, if any wound is there in the plant, that time only it will invite the agrobacterium tumefaciens, or that is the correct situation for the agrobacterium tumefaciens, or that is the bacteria to go and infect that wound. Okay, so a normal plant generally will not develop or invite the agrobacterium tumefaciens. Only the wounded plant will invite. So this is a wounded plant cell. Okay, so first thing is, from the wounded plant cell a signal will come to the bacterium. Okay, so this is the signal, that is the step 1. So next is the plant should, the bacterial cell should recognize that signal, isn't it? So second thing is recognition of signal molecules by the receptor. So this is the second step. Here you can see the signal molecule. It gets so this is the recognition step. Okay, the third step is attachment of agrobacterium to the plant cell. So here, this is the third step. This is the agrobacterium and this is the plant cell. So you can see attachment place here. Okay. So the agrobacterium tumefaciens is now attached to the plant cell actually. Okay. So this is the third step. Coming to the fourth step, activation of Vir proteins, which is a process of single stranded tDNA region. So as I told you, this is the tDNA region. So single stranded tDNA, single stranded DNA, and the tDNA complex will get warded off from here. And Vir protein also plays a very important place here, because Vir protein only will initiate this warding off process. Okay, right. So now it has come out. Okay, this is the fourth step actually. The fifth step is formation of immature T complex. So this is the fifth step. Here you can see the virulence gene as well as the T strand. So this we call it as immature complex, immature tDNA complex. Okay, this is the fifth step. And sixth step is the transferring step. Okay, so this immature complex is going to travel and it's going to get transferred to the plant cell actually via a channel, right? So this is the plasma membrane. This plasma membrane will pave a way and it's going to get transferred to the plant cell. So this is the sixth step. So sixth step is tDNA transfer, and the seventh step is assembly of mature T complex and the nuclear transport. See, once it enters, there are some proteins waiting here to invite the immature complex. So this immature complex will enter. So these proteins will support, they are all supportive proteins actually. These supportive proteins get attached and the immature complex gets converted into a matured complex. Okay. So now this is the diagram of, or this is the structure of the matured complex, and this matured complex now travels towards the nucleus of the plant cell. Okay. So once it touches the nucleus, it enters into the nucleus and randomly it gets integrated into the host cell DNA. Okay. So this is the final step actually. Once it gets integrated into the host cell DNA, with the help of the enzymes, plant enzyme, it starts expressing its own characters actually. As a result of that, as a result of that, you get your desired genes replication as well as the final output. All those things will happen after the eighth step. After the integration of the tDNA into the host cell chromosome of the plant cell. Okay. So these are the 9 steps which are very important steps happening in the TA plasmid infection actually. Okay. Right. So the next, another diagram I have just given for your reference. So this is the bacterial, this is the bacterial cell and this is the plant cell. So you can see this is the ssTDNA complex actually. This is an immature complex. You have 2 proteins waiting here. These 2 are the virulence proteins, Vir E1, Vir E2, Vir D2, Vir B, D4. So these proteins will pave a channel, and through this channel it enters, and here you have the plant cell, and even the plant cell membrane, plasma membrane also has created a channel, and very easily this tDNA will enter here, and a little bit of modification, after a little bit of modification, it's going to enter into the nucleus for its final conversion. Okay, so this is about the tDNA and TI plasmid, and thank you very much for your patient listening. Thank you.